thank you for coming to Straight Science. So Straight Science is co-produced through UAF Northwest Campus, which is what I'm housed in here, and also UAF Alaska Sea Grant. That's this position. And it's a science seminar we bring research to the Bering Strait region, especially really relevant research like tonight. For those of you who may not be as familiar with Zoom, on the bottom of your screen, you'll see like a whole bunch of little menu, if that's where it is on your screen, and you'll see a little cartoon sort of bubble, and it'll say the word chat. If you click on that, a window will open on the side, and you'll be able to text message in questions if you don't want to talk verbally. Now, tonight's speakers, Nicole and Laura Horseman, are willing and happy and would love to have a conversation. So don't hold back on the questions. Don't feel you have to. And if you don't feel comfortable shouting them out, then hit that chat button and chat it up. One thing, as we all know in the Bering Strait region, not every community is able to watch. So if we have a caller, which I hope we do, they will get priority on questions. So if we have a caller, I'll be monitoring the chat box for Laura and Nicole, but any caller calling in, we know it's tough to be a caller on a Zoom call. Can't see, can't raise your hand, can't wave. So they will get the priority. With that, like I said, Nicole Misarti, we're excited tonight. Research Associate Professor at UAF's Water and Environmental Research Center. And Laura Horseman is with UAF's College of Fisheries and Ocean Sciences. Very well known. Both of these ladies know their walrus. And of course, Laura, we also know her as a bowhead specialist as well. So with that, it's in their bones, walrus past and present. We're looking forward to hearing kind of how walrus have been doing and where they are headed. Take it away.
Oh, all right. Thanks, Gay. Thank you for that introduction, and thank you for having the two of us. We're super excited. We've been trying desperately to find ways to get out to see all of you, and of course, with COVID, we can't. So I actually want to thank Vera too, because she was the one who said, "Why don't you just go ahead and?" Oh, well, that's interesting.
What?
It's not advancing.
Oh,
so we thought we were going to have no things, but Vera's the one who said, why don't you go ahead and come through Straight Science and talk to us through that. And so I thought that was a great idea.
Straight screen.
And there we go. All right. So the first thing I want to do also, besides thanking Vera, not only for that idea, but also because she has allowed us in multiple years past to tag along to some of her meetings in Gamble and Savunga, but also to everyone that contributed samples to this project and all the knowledge that people passed on when we were able to travel and talk to all of you. So that was sort of the first round of thank yous that we wanted to have. We are a huge group, tons of students, most of us at UAF, not everybody, but some people actually in Nukiavik and Barrow, and then students that came through the Rural Alaska Honors Initiative. So that was really fun to host them. We had them for a summer, and some of them made posters and they traveled around.
And Michelle won an award.
Michelle won an award when she was at the Alaska Marine Science Symposium with us. So that was really fun. And we'll show you some of their work later. But, we couldn't have done this also without all of our collaborating entities, the Eskimo Walrus Commission, U.S. Fish and Wildlife, the Alaska Department of Fish and Game, North Slope Borough. This was really a massively collaborative project, and it's kind of been going on for years. We've been a little quiet the last 2 years because we're mostly at the end of what we're doing, and We're
And
hanging in COVID, right? And we're hanging in there in the hopes that COVID will lift enough that we can come and talk to everyone one more time before we go.
But key thing too is that it sounds like this is work that we've actually done. And in reality, it was a lot of really amazing students. And I think you all know that. Some of you have met them. And so especially Patrick and Casey, who did amazing, amazing work that we have the honor to present today.
Yes, this is very true. And, you know, we started out thinking, well, we have some pretty important questions about walruses, and some of them are pretty basic, but we really wanted to know about them. How resilient were they to climate change? How has their diet changed over the last 2,500 years? And actually, we were able to push that back to almost 4,000 years. Since we started this project. We wanted to know if that diet changed in predictable patterns. Did we see changes with sea ice? And as we were working along, we also decided to look at whether or not teeth could serve as biomonitors, and could trace elements and hormones in those bones and in the teeth predict population size. And of course, population size is something else that we could look at changing through time, and sort of see if that fluctuated with sea ice, without sea ice, and just how all of that worked in the past. And as everybody who's on this call pretty much knows, there's a lot of possible stressors for walruses as we're facing climate change, right? And they can have adverse health effects, including decreases in reproduction because the energy is going elsewhere, stress based on nutrition, or making up energy efficiencies because they're swimming farther from land to forage. There's stress in hauling onto land in large groups rather than slightly smaller groups spread out on the ice. So there's all those stressors that everyone's well aware of, and we were just hoping to see how walruses may have handled this in the past.
And now,
yeah, because we looked at now as well. So here's the weird thing about us and our project. Most of the world sees things as fleshy beings walking around, and when I look at things, I see them more like this. I look at things and I worry about what's in those bones, what's going on. And I can tell you there are a million things that we can pull from the bones. And the science is only getting better. It's really geeky science, but the basics of it are that we can pull hormones out of bones, both for reproduction and stress. So we can figure out your diet, the productivity. We can look at, just with even simple measurements of bones, size and sex. We can look at metal and contaminant uptakes into bones and into teeth. We've learned age at sexual maturity can be figured out through teeth. We can figure out where these walruses were swimming. We didn't wind up happening to do that, but this can all be done with teeth. And of course, DNA is something that people talk about in ancient DNA. And we can look at population size and diversity and identify populations over time as well, looking at all of that. So the bones are actually, and the teeth are actually way more information than anybody would have thought of 20 or 30 years ago. This is all pretty new science. And so they're becoming more and more important. I always just try and let people think of it as it's an archive of the past, right? It's an observation system. It's just a different type of observation system than you think of. This isn't a buoy out in the water taking sea surface temperature and other measurements, but there's so many measurements that can come from this. And because Alaska has all of these amazing archaeological sites where people used to leave their remains of their dinners and their trash behind, we can access that. So even those trash heaps are really incredibly important, and are little windows into the past. So we'll start with trace elements, and we're going to talk about that and about how we can look at those through teeth. So the background, as far as we're going to give you, is that teeth, like tree rings that you see down at the bottom here, grow incrementally. And they're little biological archives that you can look at from year to year to year. And so we looked at a bunch of trace elements in walrus teeth, and we found tracers of petroleum-related activities and also markers of biological milestones like weaning and maturity. So the first thing we did is pull teeth out of mandibles or jaw bones if they weren't already free floating around in what we were doing. And then this crazy little saw there slices this super thin layer. It smells a lot like being in a dentist's office.
Yeah,
it's not
horrible.
the best smell, right? But it does make a beautiful tooth section, and that's what a tooth section looks like. And so there are different parts of the teeth, and we were mostly looking at the cementum, the lifetime, right? And that grows from the inside out. So the inside's the younger layers, and the outside, the last layer is the layer that was put down just before the animal died.
Can you point at it?
Oh, can you guys see my pointer? So we're talking about birth basically here, and the death rings. Excellent, thanks, Gay. The last year is out here on the outer edge. It works just like a tree ring here in the center versus the outer point, and we can count them just like we can count the dark and the light layers in a tree ring, right? And so this is the this is the top. I always do that, the crown. So the tooth, part of the tooth you can see, and then this down here is the root, the part that you can't see that's embedded in the jaw. Then we take that nice slice and we actually, it's this very fancy instrument, and we throw a laser at it, and the laser beam comes down and hits it and shoots off all these little atoms and particles and all of the rest of it, and we can read those. And we read them for particular elements. So in this case, we looked at zinc and lead, just to look at what happened. But you can see from moment one here, this is your slice. This is the actual scar of the laser going across all the different layers. You can kind of see the layers in there, and we've painted them out here for you. So this particular walrus was 10 years old, 0 to 10. And then we could take the data that was shot out from the instrument and overlay it over those scars, right?
And we're getting more into this in a minute.
Yeah. So one of the first things that came about was looking at vanadium, and vanadium can be a crude oil tracer. And we can actually see that there were peaks and troughs in how much there was. And it's a way to actually monitor that all the way into the past. In this particular, we only went back to 1945, but we could potentially go back farther as well. And these are all male walruses that have the big spikes, which is also interesting. The purple's the males.
Purple is male.
And the greens are the females.
Yep.
So you can even see in some ways the discovery of Prudhoe Bay and the use of that starting from the late 60s. And there are a couple spikes, but the vanadium as a tracer went back down, and then, there's different times that you can see different spikes. And we're not entirely certain if that is just because of where these males were feeding, where they were staying at the times, in these particular times in the 70s, the 80s, and in the early 2000s, or if it was just actually what they were eating, because oddly enough, sea peaches or tunicates actually also have vanadium, high levels of vanadium in them. So there's also a very small possibility that these guys were eating a lot of sea peaches, and that is what we're seeing in there. But more than likely what we've got here is a crude oil tracer. It's really cool that we can see this all the way back into time. Cadmium is a potential element of concern because it's not exactly healthy to have high levels of cadmium in your system or in a walrus's system. And cadmium accumulates, which means that over the lifetime of a walrus, and you can see it right here, it increases in the teeth, right? But what's most interesting is that the source of it has either stayed the same or decreased a little bit since 1950. So cadmium, for whatever reason, yes, Gabe.
You're muted.
You're muted.
Happens every time. You think I'd know by now. But anyway, just in the spirit of the, you wanted a conversation. You have a question in the chat from Rick Toman, and that is vanadium a tracer for crude oil specifically, or other things like diesel?
Well, other things as well, including prey. So like Nicole was saying, the vanadium is actually an essential element for sea peaches. So as we are using in our blood, we're using iron to bind oxygen. Sea peaches use vanadium for whatever reason. So it's entirely natural, and it can be a tracer for crude. It can be a tracer for drilling muds or for drill cuttings. Pretty much a lot of these kind of things.
A lot of things that are associated with oil extraction, it is associated with, but then there's also the whole sea peach story as well.
Right. And so different crude oils actually have different levels of vanadium. So some people can, on the amount of vanadium that is in crude, can actually tell where it's from. Not us, but yeah, it's possible.
It's a natural element, yes?
Yes.
Yep.
So sea peaches or any kind of tunicates, those are walrus food.
Yes.
Walrus, they like to pop them like a grape.
Yep, exactly.
And you'll see them in the stomach. So, and sea peaches are good for people to eat too. So it's a food here that's targeted. So it's interesting about what you find in the sea peach and in the walrus. I wonder if you would find that in people who consume tunicates on a regular basis.
Possibly.
And it's possible. Yeah. Yeah. I mean, it's made it all the way into the teeth. So it's definitely moving throughout all parts of the body. So
it's being deposited.
And there seems to be in the bone.
In the bone. Yeah. In the tooth for sure.
This is in the tooth.
This is in the tooth. Yeah.
Okay.
So,
interesting.
All right. And yeah, sorry, the chats, they show up as a little bubble for me, but they would be covering my slides that I'm showing you guys if I left them. So keep interrupting us.
Not a problem. Don't worry about that, because that's what I'll just keep monitoring. And since you wanted questions right up, I'll just keep busting in if people have questions like that. I'll let you know. Don't worry a thing about it. Just keep going. You're doing great. Lovely graphics.
Thanks. Oh, and then every time I check, it does that.
Okay,
so
go back to the cadmium for 1 second. So cadmium, and I think a lot of you guys have actually heard about cadmium when Lori Quakenbush was out there and did a lot of sampling of walrus cadmium. And she also found that compared to the 80s, cadmium actually went down in the tissues. And if you're looking here at this graphic, which we can't see right now because you guys are all over it, but you can see too that over time it actually has gone down, and that there was a little bit of a peak here in the 1990s, and then later on cadmium actually kind of went down. So, and that's something that actually we did find in, or Lori did find in the walrus tissues as well. So I just wanted to mention that real quick.
And it's decreased quite a bit since the 50s, which is also of interest.
Yeah.
All right, so this actually, so we started out, was a happy accident. We started out looking at contaminants, trace element tracers in the teeth, just because there was a contaminant project that our student, Casey Clark, was doing. And in doing this, he ran so many teeth that he actually started to see some patterns. And this was this happy little accident pattern that is really fun and amazing. And it has to do with age at maturation, which is also a measure of population productivity, right? So I'm going to let Laura talk about The ovaries, and how we validated that this actually works, because I do it wrong every time.
Okay, so before we're going to show you the actual patterns, this does relate to female reproduction a little bit, and I just wanted to give you just a smidgen of a background that we just didn't pull that out of our behinds here either. So we actually validated that with, female reproductive tracts that were matched together with teeth. And so one thing that you can see up here is actually a cross-section through a walrus ovary. And I hope you can see two structures. So one is this yellow blob that has a little bit of black on there. And then there are these two white blobs over here. And so when a walrus is ovulating, and that normally leads to a calf, to a baby, this ovulation then becomes this yellow blob right here. And we're going to hear a little bit more about the hormone that maintains the pregnancy. But this is what this structure does. It creates this hormone that maintains the pregnancy. When the pregnancy is done and over and gone, this structure is recessing or kind of decreasing in size, and it becomes these white little scars. And what we can do now is we can actually count all of these scars in both ovaries from a walrus, and we know how often that walrus has had a calf in the past. And usually we kind of assume a 2-year interval between calves. There's a little bit of variability there, but that worked really well. And so essentially what we get by looking at this is have a really good idea of what the age of reproductive maturity was when we know when the walrus had died. So what we found then was in the teeth, and what all females kind of ended up showing very, very strikingly, was a very low zinc levels that then all of a sudden increased at a certain point in time. And so we kind of saw this change point, this, where that actually happened. And when you think back on how we overlaid this on the teeth, this change point was actually the point where the female started to enter sexual maturity. So we had now a way of going through all the teeth that we looked at and look at sexual maturity in walruses through time.
Yeah. So why is that important? That's basically important because you can actually look at reproduction based on increases and decreases in population and competition for mates and competition for food, right? So if you have a smaller population, there's less competition, supposedly more food out there, and it tends to be that there is a lower age of reproduction. They start reproducing earlier in their lifetimes. But if there's a large population and a lot of competition and not as much food to go around, then that age of reproduction tends to rise. So based on this sort of ecological know-how that goes for more than just walruses,
yeah, this is actually something that is well known from humans as well. So during World War II, when people were starving, the age of sexual maturity for humans was actually kind of 15 years or so. And if you're looking now in our population, at least in the US, where everybody is well fed, sexual maturity can be as early as 7 to 9 years. So you can see the exact same thing as well. So food and sexual maturity are very much related.
So what we did was we went all the way back to 1910 with these teeth. So we have about 100 years worth of data here. And we looked at this in relation to what we know about population size based on records from the past. And so when you're reproducing at a younger age, your population is low, which it definitely shows right here in the early 60s. But then as the population grew in the 70s, that age, which is represented in these boxes, increased. And then when population came back down, starting in the 80s, that age started to decrease as well. But what's really interesting is our data from 2005 to 2009, which actually has a pretty high reproduction, and supposedly the population's not really that high. So the question is the carrying capacity out there right now, the carrying capacity of the Bering, and is probably a little bit lower, is what we're thinking. Now we have a question mark after that because we don't know for sure.
Well, I think nobody really knows what the population size, right, is. But keep that in mind, because we're going to come back to this, because we have another line of evidence that shows us exactly the same thing. So it looks like right now, based on the data that we have, is that we have a very strong walrus population out there. But it could also mean that the ecosystem, because of the change that we are seeing, the ecosystem can just not support as many. So it would look like a big population, but really it's less food.
So here was the other happy coincidence. We also were able to find an age at weaning, which is, as most of you know, an indicator of health of the mother and the calf. And yes, I get to say health there. It's the one time I'm allowed to say health out of this entire discussion. But of course, that portion is actually really complicated, because if you're weaned early, it could be because there are not enough resources for the mother, right? And it's also involved with a lower calf survival rate, right? But it could also be that there's an increase in food availability for the calf, so the mother can wean the calf earlier because the calf is going to be okay, right? And then that later weaning, it could be that there's a decrease in the predictability of those seasonal resources. But a later weaning is also related to actually an increase in the survival of the calf. So it's really complicated, and the timing of all of it is also related to whether or not another calf comes along, right? So there's something in the chat, and Gay, I can see your face again.
Yes. And so Rick Toman had written in the chat, he was, I think, commenting on, age of maturation.
Yep.
Right. At 2005 to 2009 was prior to the most recent environmental shocks.
Correct. Yes.
Oh,
correct.
Yes.
Yeah. Mm-hmm. And, and this is, as far as we are concerned, it is really not a good time to stop this kind of work. So that's kind of unfortunate, but that's definitely something that we have on our radar, because things, and you'll see this in pretty much everything we're going to show you, is things are really weird right now.
Yeah.
They're really weird and really strange.
We have 4,000 years of data, and things are really weird right now.
Yeah.
Walruses up to the point where we were able to look at them seemed to be doing okay with it, but things are really weird right now.
Yeah.
All right, so in order to look at that age of weaning, we looked at strontium and barium, which are found in the mother's milk in really high concentrations. And so you can actually see here, again, a change point as it comes down, right? So we've got the same exact thing. We've got the tooth. We run the laser across it. It shoots us out all sorts of interesting data. And we lay that over the age, the tooth, so that we can see what age these changes happened at. And this is what strontium and barium sort of tell us about weaning. You don't take up too much strontium and barium as an adult, because there's discrimination against it in your gut, and there's also a little less of it in the diet than compared to, milk, but we'll get to that in a minute. So then the mother, passes along what very little she has, but then when the calf starts in the placenta, when the calf starts to nurse after it's born, there's a lot of that strontium and barium in the actual milk. Plus you've got an added thing here, and that is that your gut doesn't develop that strontium or barium discrimination until a little bit later in life. So you've got two things happening at the time of weaning. You're losing high intake of strontium and barium, and you're developing the ability to discriminate against it as well. So, and here we have, you can see this is barium concentration. You can see this is where we would have called the change point, right? It's been coming down at the end of weaning, and then this is the rest of the lifetime. And you can see that here, and we were able to overlay it. And you can see that happening here at around, 2 to 3 years of age, right?
Which is actually something that Bud Fay kind of suggested, that most of, and most of you know, I mean, he's the walrus god from the 50s and so on. So he wrote, kind of the book on walruses. And so he suggested aging, or the age of weaning for walruses is around age 2. What is interesting is that based on the data that we got, actually that wasn't always the case. And so the Fay idea here of at age 2 you're being weaned is in that gray box right here that is laying behind.
So each one of these colored columns is half a year.
And but you can hopefully also see that a lot of them, so this is how many animals there actually are. So we have about 50 of the animals that we're looking at that were weaned at age 3, 4, in some cases even age 5. So we have definitely a way of looking at this, and that it's not what we really always expected. So some of these calves are nursing for a whole lot longer, which is a kind of a measure of calf survival. And one thing that we haven't done yet, what is going to be really, really interesting, is look at this through time. So is that something that can tell us something about good times and bad times in the ecosystem at that time as well?
Yeah. Okay, there's something else in the chat here. Let's see. Oh, no, never mind. It was an old one. All right, so we're going to move from that onto steroid hormones. And so we're leaving the teeth for a minute and we're going into the bone, and we're looking at reproduction and stress hormones with this. So we looked at 4 different hormones. We looked at cortisol, progesterone, testosterone, and estradiol. And they all tell us slightly different things. So cortisol is related to stress, and progesterone is related to pregnancy. Testosterone is related to male sexual behavior, and estradiol is related to sort of the body preparing for pregnancy and, being in the midst of pregnancy. And so all of this together can sort of help us look and identify population physiology through time.
And that may all sound pretty boring right now, but actually what hasn't been done before we did this is it has never been extracted from bone, especially marine mammal bone. So this was kind of a very novel thing to do, and it actually worked, and we got some amazing data out of it that really was quite fascinating. Yeah. So
we were quite pleased when it actually worked. We had set it aside as a smaller part of the project, and when it worked, it became pretty important. So again, hormones can help us actually measure population size, in the sense that a smaller population plus means increased competition for finding a mate, even though there's increased food. But it could be an increased stress and an increase in pregnancies, whereas a large population is a decreased competition to find mate, and then but potentially a decrease in the availability of food. So you might decrease the stress versus the higher competition of finding a mate, but you're also going to probably decrease pregnancies because of the decrease in the food availability.
And also for the female audience here that have born children, you all know how stressful this is. So pregnancies and going through a pregnancy will definitely increase your stress level quite a bit.
And apparently mating for the males is
also very stressful.
Very stressful.
Yeah.
So we did look at the hormone levels through time, and we found that low populations, and I'm going to put the population curve is in red, and I hand drew over it so you guys could see it really well. And the actual data for cortisol and progesterone is in black. So you can see when the population is low and when it's high. And when the population was low, there's higher rates of pregnancy right here. Sorry, populations are low, pregnancies are high based on progesterone, right? And that inverses later. When the population goes up, the progesterone for females comes down. When you look at cortisol, the stress, and this is mostly driven by the males in our study, when the population is low, you have a slightly higher level of cortisol, and when the population is high, you've got the lower level of cortisol. Is that correct?
Yes,
I'm telling
so,
you. Right.
but one key thing to look at right now is again our 2010, 2011 kind of data. And hang on, I gotta move this. Yeah, we're gonna move you over there. So if you look right here, so this is our pregnancy data, right? So this is what's happening right now. So this indicates that there's actually very few calves being born lately. And that would again indicate a large population, which is very, very similar to what we've shown you before, where we actually found the age at sexual. I'm not sure. Okay, your computer is going crazy. You got a
Don't
pointer.
touch it.
Where we found the exact same pattern. So I'm gonna go back to the.
So the question is this really a high population or just a lower carrying capacity? In other words, there's less resources out there for these walruses, or the less ability to get to those resources.
So another really important point to make here right now is that in the lower graph, this is going to show you a measure of stress. And you'll see they're really not stressed. This is very, very similar to what we found in the 1880s. So they were kind of a higher level of stress, again, in the 50s when the population was tanking. But what we are seeing right now, which is really good news, is that the stress level that we were concerned about with regard to reducing sea ice and so on has not manifested in the bones anymore.
As of 2010.
As of 2010.
And
Correct.
what was interesting that we didn't show here is the levels of the cortisol levels that we find today are the same also to the cortisol levels that we found even before the Industrial Revolution and before sea ice even started to get affected at all by humans.
Question from me is, so the way I look at this, your population is only up to 2010, so that's 11 years ago. So we've got a lot that's happened between them, but and it's really interesting stuff. So the red is actual data on population, and the black is your the first one is your hormone. And my question is on the cortisol. So that's really interesting to me. Why is it that when you have really high populations, you have such low stress?
Well, we're wondering if this was also driven by males in part, because there are a lot of females out there and there's not a lot of competition for mates and harems.
Yeah.
And
so everybody gets to breed.
Everybody gets to breed, and for whatever reason, that is less stressful.
So these were mostly male.
This was driven by the males when we looked at the cortisol was driven by the males.
Okay, all right, yeah, okay. Because I the picture's like a mom and a baby, so I was like, oh, that's
right.
Really
that was progesterone.
Yeah, okay.
And there's female in there, right? So this is not just the males, but a lot of the patterns that you see there with the low versus high population it was driven by males.
Yeah,
and that makes sense to me there. Thank you. I was like, I got it. A lot big, yeah.
Yeah. And what you were saying about, yeah, this ends at 2010. This is why Laura and I keep saying, like, this is not the time to stop this research. So there's 11 more years of teeth available out there that we could be doing, and bone that we could be doing a lot of interesting work on.
Yeah.
And so this, again, was just to show you that we are finding the same odd pattern at the end here. And then we were going to move on to isotopes, which can tell us all sorts of things, but we're mostly looking at diet here when we're looking at the isotopes that we're looking at. So I put up this all the time. It's the stable isotope food web. And the reason I say it's the stable isotope food web is because it's not going to be like any other food web you guys are used to looking at. It's based on the chemistry. And that's because you absolutely are what you eat. So here's higher trophic levels at the top, or at the top of the food chain. And at the bottom of this graph is the bottom of the food chain. But we also have, from left to right, things that show that you've been feeding more in an open ocean water realm versus more shelf waters and bottom feeding, benthic feeding types of things. And you can just see it move up from the bottom and all the way up to polar bears. This is where walruses are. The reason they seem slightly over to the side is because they are benthic feeders, so much more so than any of the other creatures and seals and stuff that we just showed you. And if you are a human being in Kotzebue and you're eating a little bit of, both food from the store as well as subsistence foods, that's about where you would land. So the first thing we are going to talk about is the whiskers. We did not just bone, but also whiskers. And I'm putting this in here for two reasons. One, it matches up with tooth data that we'll tell you about, but we won't show you. And two, because we were so excited. This was all the Rawhide students. So this was all of those students that presented their poster, and it's really cool data.
And keep in mind, those are high school students.
Yeah.
So those were high school students, and they did amazing stuff.
They came in, they learned about isotopes, they did lab work, they ran data, they analyzed it with us. It was great.
Yeah.
So what we have here is two different females, one in the top graph A and one in the bottom graph B. From Gamble. Both of them collected, one in 2015, one in 2016. And thank you again to all of the hunters that collected those for us. On the left side of the graph, you have the base of the whisker, and on the right side, you have the tip. So what you're looking at from left to right is the most recent on the left, and the older stuff 1 to 2 years ago at the tip. And in the top graph, we have a female who was clearly eating fairly high on the benthic portion of the food web.
So scrabbling down in the mud.
Scrabbling down in the mud and getting not just clams, but a lot of crabs and other things. And we can tell that based on both of these different isotopes, both the nitrogen and the carbon. Right. But then at the beginning of this 1 to 2 years ago, she was clearly eating clams, because you can see this separation that tells us based on the isotopes that they were eating something clearly in the benthic realm, but also very clearly pretty much the bottom of the food web.
Yeah. So this is super, super low in the food web and clearly in the mud. Yep. So it stayed up in the mud and very low on the food web. So this is a classic clam signal.
Yeah.
So, and then here something very different happened, because the exact opposite happened, right? So you can see here, it was kind of low like this one was, and carbon signal right here.
So eating clams.
So it was eating clams until 1 or 2 years ago, when all of a sudden we have very, they look like polar bears here. So they a very high trophic level, and all of a sudden a very, very low carbon signal. And this is classic for a seal eater. So this is very, very typical signature of eating essentially something that's very high in the trophic food chain and full of fat. And a seal fits that bill.
Yeah. And we ran 5 females and 5 males total with the rawhide students. And we found really variable foraging. So their diets were all over the place, eating all sorts of different things. But what was also interesting to us is that you could see sort of when males and females were in the breeding areas at the same times, because all of a sudden their foraging behavior looked really, really similar. And at other times, when they weren't in those breeding areas in the same place, they looked really, really different. The other thing that we found surprising was that out of those 10 seals, two of them, or sorry, seals, sorry. Out of the 10 walruses, 2 of them were eating seals. So if that actually extrapolates to the rest of the population, and we don't know whether or not it does, but that would mean that almost 20% of walruses have seal in their diet at some point in the span of, 2 to 3 years.
And that actually
Super
fits surprisingly well with an early student of mine did work on muscle tissue, and she found pretty much the same thing. So that walrus diet pretty much was 20 to 25% higher trophic level stuff. So quite fun and interesting data.
Yeah. And we don't have we didn't show this, but we actually have the same story from the teeth. So we have 29 individuals with the teeth. Yeah. And we found that same thing, that same 20%. Pretty much, which we were shocked at. We thought it was a really rare thing. Okay. Moving on. So then we took a really long-term view. Oh, yep, Gay.
Yep.
We do have a question in the chat, and that is, what would the signal be for fish? And you probably can't see this one. This came right into me.
So it depends on where.
On what fish?
On what fish. So likely for the fish, it could have a somewhat higher nitrogen depending on which fish.
Not as high.
But not as high. So somewhere probably, in here, 15, 14, something like that. And the carbon, depending on whether or not they were, so like if it was like a kelp forest fish, the carbon would actually be pretty high. But if it's a super pelagic feeding fish, then the carbon would be fairly low, not quite as low as a seal, because a seal has, there's 2 things going on that's driving its carbon signature, and that is the fats as well as where it was eating in the system, right? So the fish is only where it was eating in the system. It doesn't have quite that amount of blubber that's driving the fats down.
I mean, they're lipidy, but not like a that. Seal. Not like that. So it would look a little different.
Yeah. So to sort of try and make it, these 2 points here might come a little bit closer on the graph.
Yeah, it would probably sit.
Right? Does that make sense? Is that clear enough for the audience? It's so hard when I can't draw it out for you.
Looks like it. I don't see anything that says no. So thank you for that.
Okay.
And then my question is on this whisker, because I'm always interested in the whiskers. That says tip 1 to 2 years ago. That's the growing rate. You're not actually talking about these 2 animals these are results from 2019.
Oh, yes.
Oh, you're
okay. No. You're absolutely right. So one was harvested in 2015 and one was harvested in 2016. So what you're looking at is 2015 here on the left side of this graph, and somewhere around 2013, something like that.
There you go. Thank you.
And you're welcome. All right. So then we took an even longer term view of walrus diet through their bones. And we collected this through archaeological sites, museums, and everything else. This is, Rick's going to hate this probably. This is a super simplified idea of what has happened in the past. And what we basically have, the most important thing to take away from this is we've had warm periods in the past and we've had colder periods in the past, right? And we are back into a warm period that's beginning to spike pretty darn quickly and getting up there to be some of the warmest times we've had in many thousands of years. But it's not completely unprecedented to have had warmer time periods in the past. And this is important because if we're losing sea ice now during this warm period, we likely lost some sea ice during other warm periods as well, just like we had greater amounts of sea ice during things like the Little Ice Age. So what Casey found in these bones was that, oops, I'm trying to move you guys around so I can talk to you about this. What he found is that we do have changes through time from 4,100 years ago, which is the oldest bone we had, up until 2016, which was the youngest bone that we had available to us. And what he's basically found is that the carbon tells us that walruses shifted to sort of less diverse diets over time, over the last 4,000 years, right? So that's this carbon here. And you can see that the diets were a lot more diverse than they are either today. I just realized
you see it on the nitrogen too. So both of them together.
Yeah, you can see it both in both of these things, these diverse diets. And today it is a little less diverse and it's grown and contracted over time. And that's really the most important thing because diversity in diets is what's helped these walruses make it through all of their, all of these changes in the past. And there is the potential coming from this that there was slightly more variability in diets during periods of low sea ice than there were during periods of high sea ice. But that didn't quite hold for the moderns. So we're not quite sure that
are they getting to that point? So are they going to look, in the next 10 years or so, are our modern samples down here, are they going to look like those from 4,000 years ago?
If we keep sampling.
So are they going to diversify further? Or are we completely different now than it has been in previous low ice periods? So those are, diversity is good, right? If you are eating a lot of different things, you don't have to starve. But if you are only eating one thing, like panda bears, right, you are in trouble. So in this case, kind of becoming narrower and becoming more specialized in what they're eating is not good. So we are not entirely sure what's going to happen, and that's yet another reason why this is not a good time to stop doing this
kind of work. And what to us is also really interesting is the orange here, the nitrogen, again, that's position in the food web, has dropped quite a bit over time as well. So this whole reliance on clams that we think of as being this massive part and typical of walruses may have been a little less typical in the past. So that is interesting to us as well. So the last thing we want to talk about, and we'll try and speed it up because we're really getting close here, is the DNA, which we looked at for population size and diversity, right? So it can give us these estimates of population size. It can also identify genetic bottlenecks, like you see here, some catastrophic event, and then, an expansion again. And so in our case, we were actually looking to see if that massive commercial walrus hunting that happened at the same time that we had all the commercial whaling in such a big way made any difference in the 1880s. And it turns out that it sort of did. So this is a very complicated and typical DNA diagram, but what's important to take away from this is that some of the past genetic markups that we have are actually no longer present in modern day walruses. And that's all of this purple, right? And so anytime you see purple in its own little circle, that means it didn't carry on into our modern day Pacific walrus population, right? And when you run all of the data, there actually was a small bottleneck during the commercial walrusing, and that then re-expanded, but we don't know if these particular genetic markers were lost during that bottleneck, or if we just don't have enough of a modern sample to have found it in the modern gene pool. And I'll tell you, this is not just our DNA work, but there also is a big bank of data called GenBank, Genetic Bank. And we took all of the walrus data they had from that as well for the modern stuff to look at that. So what we found in the modern and what other people have found in the modern Pacific walrus populations is missing those few genetic marks that we had earlier. But the diversity expanded in a really big way once again after walruses came back. So it's not that we lost diversity, it's that we may have just lost certain little genetic families.
We lost some families,
but we gained others. That's a great way to put it. So our big point from there is diversity is resiliency, right? It's usually populations that cannot keep the genetic diversity going that really begin to have problems. So we're not going to say too much more about that because, Gay's got a question, or a time.
No, time is irrelevant in the Bering Strait region. We like learning about walruses. We really do. So don't worry about that. But you have a question that was texted to me. So that's novel. And that is there meaning with this figure? And I'm guessing it is this figure. So whoever's texting, go ahead and say if it's this figure. Is there meaning with this figure? Measuring thousands of and hundreds of years. Oh, the one prior. Your prior figure.
This one.
Yeah, I think so. Is there meaning with this figure measuring thousands and hundreds of years, now only 4 years? And yes, this is the graphic.
Yes, this is the graphic. And there is, and this is, we chose one graphic because it makes a good picture for us to look at and talk about these things, but we actually did
more.
We have more that actually shows that we do indeed have this slight loss. Because when we're looking at things, we actually did break it down into smaller time periods.
Mostly historic.
Historic, and it tells us the same story. So yes, that is a good thing to note. We're looking at 4 years versus a couple hundred in each one of these, or a couple thousand. But when you break it down as samples per time periods, we get the same story.
Yeah.
So yes, thank you. That was a good question.
Okay.
And then, so we're going to end it pretty much with the story that diversity is keeping these walruses, these walrus populations
in relatively good shape. Don't say
healthy. I was going to say healthy.
That is the one thing that you can't tell from bone.
Evidence, basically shows that we have potentially either a high population size now or changing carrying capacity. And we're looking at that through age at first calving, which is the zinc and the hormone data. And we've looked at high diet diversity through whiskers and teeth in the shorter term and bone in the longer term. And that diversity, we think, has definitely helped walruses survive when their ecosystem is changing and when the sea ice is going away. And we have higher diversity in the weaning age than we originally thought. And oftentimes it's later than what we've previously known. And that could help with higher calf survival. But that ability, again, to adapt, adjust, and be more diverse is definitely helping out the walrus population. And again, we have high genetic diversity as well. So that to us was one of the takeaway messages of this, that although we need to do more work to see what's been happening over these last few really extreme years, for 4,000 years at least, these walruses have navigated quite a few changes, and they've done it pretty darn well. And you know, it also comes down to resiliency, right? That's what we're all worried about. Are they resilient? And we think they're fairly resilient. Now, again, as we move into sort of these unprecedented times, more work needs to be done to see just how resilient they're going to remain over the next decade or more. But we've developed all these new ways with this project to sort of determine age at weaning, age at reproduction, stress and sex hormones that we can compare over thousands of years. And all of these tools can be used for management and decision-making. And we're also hoping to apply many of them to other marine mammal populations that are also important to everyone, you know, living on Bering coastline. Right.
So, and you've seen, you know, some of the teeth that we've used. All these are pretty big, and I would say the biggest limitation that we have right now. But Nicole and I have a plan. Is that it would be much more difficult to do on a very small tooth. So if we wanted to expand this work to you know, ringed seals, bearded seals, spotted seals, and whatnot, this would be difficult. But again, we have a plan on how we could apply this. And this is the main reason why this kind of tooth work in the past has been done with sperm whales, where you essentially have a tooth that is the size of a basketball.
Yeah.
Anyway, it's a little over exaggerated, but
so you can I put my email down there. You can contact me with any questions. I wanted to also say we have a ton of other things that we've done with this project that we just didn't have time to go into tonight. And one of those things is that we transcribed and digitized a lot of old interviews that we found in the library. And we gave some of those out when we were visiting Gamble and Savunga in the past. But what we want to do sometime over this year is put together jump drives that have presentations like this, that have all of those transcriptions as well as the digitized recordings, so you can actually listen to them on those jump drives that we can send out to everybody, as well as the very geeky sciencey papers. We'll put those on there. Many of you will not want to read those. But we're going to sort of put together this package and put it in a way that we can easily send it out, and you can just plug it into your computer. You don't need, great connection at the tribal council. You can do it. You don't need a fantastic connection. You don't need to hear us or try and, Zoom this kind of video. It's going to be there. So if you're interested in that, then please do contact me. And we can get one of those out to you. And I think that's it. The rest is just, we had a ton of funders. And again, thank you to everybody that helped us make this project possible and that was willing to sort of, do the odd thing, which I know was weird for many of the hunters. They're so used to ADF&G and US Fish and Wildlife asking for whiskers and pieces of blubber and skin. And we said, by the way,
give us a bone.
Could you give us some bone? But thank you for doing that and putting up with that.
All right. Well, there's actually two things going on at once. You still have a question in the chat box, which we'll get to. And the straight science audience knows what to do. We always like to give a little love to the speakers because it is not easy being a straight science speaker. You're taking your time in your evening to take care of us. Gnome Nugget's giving you a clap. I don't know if you can see that.
Yep.
So
yeah, thank you.
It's a very positive crowd, very supportive crowd for any of the speakers. And the question is, what sample size for teeth would you need, not just old from midden sites to current teeth, especially when looking at smaller marine mammal populations in thousands?
So when it comes to teeth, we actually need a whole tooth because we slice that in half and then we do that whole slice down there and we run the laser across all of those outer rings. So we actually need a whole tooth or half a tooth, but it needs to be the full portion of the tooth.
Right, but they are asking how many teeth would you need?
Oh, how many?
So I think, right? That's the question.
I see examples.
It says I meant I meant the number of whole teeth.
Yes.
Yeah. So we were able to see these patterns in walruses in, kind of as few as 10. So, it's like the more you have, the better, but I think 10, 15 per decade, for example, or right now per year would be fantastic. If we wanted to keep monitoring this, I think we can make, kind of continue seeing these patterns with 10 to 15 teeth.
Wow. Okay. Anyone from the audience have any other questions?
I
Or
have
either
a
Nicole.
question, if that's okay.
Yeah, absolutely. Go ahead.
So one of the things that the Intergovernmental Panel on Climate Change is sort of predicting is that in the next 30 or 40 years, we won't really even be seeing sea ice. How might that impact future walrus research?
Well, I mean, the research, it shouldn't impact because we can continue to do our research. And if you look at Atlantic.
Well, it depends. If the hunters don't get walruses
well,
anymore,
that's
we
true.
are not going to have.
That is true. That depends not on the research or the researchers. It depends on being able to get to the walruses. And so that's going to depend on the conditions and adapting to conditions for hunting, which we know is not going to be easy, right? That's a factor in this that, doesn't necessarily have to do with the walrus population, right? It has to do with the ability to access them. So all things being equal, if we can have access to those, it won't affect us doing our research, but it's definitely, with no sea ice, going to affect walruses, right? I mean, look at the Atlantic population of walruses. They're much smaller. They haul out on land. They don't rely on sea ice the way the Pacific walruses do, but they are a much smaller population, and they have bottlenecked and definitely lost genetic diversity over the last while. So there's no telling.
Yeah,
where
I guess I'm not quite as pessimistic on this. So I think because of the Atlantic walrus population, being very happy without ice, I think the Pacific can adapt to it as well. The key problem, I think, with climate change, and we've seen they've done it in the past, right? We've had warm periods. They've done it in the past. They came out okay. But the issue, I think, is the rate of change. And so can they really adjust their life history that quickly? And I think that is something that we need to keep an eye on and need to continue to look at. Yeah.
All right. You're getting a clap from Peter. Julia, did that answer your question?
Yeah, definitely.
All right. Any other? I've got a couple questions, but I'm going to wait. Give everyone first cut. Anybody else? Peter dropped off, but I don't know if you saw he was giving you hearts and.
I know, I saw that.
Yeah, he was enamored with it. That was great. So for me, most interesting, we're seeing all these signs of food availability problems in other parts of our northern Bering Sea, Bering Strait region, and probably we'll find out more this fall when people are going to be talking to us about the cruise results here on Strait Science. So we're excited about, we have kind of a good lineup for that. So that what you're looking at is, or what you're telling us is sort of going along with that very vague hypothesis that there's a issue with food availability, perhaps, or carrying capacity overall in the Bering Sea. So that's interesting to me. And thank you very much for, I mean, this is a huge project you guys are doing. And because we live in the Bering Strait, I was just wondering which communities and sort of the bulk of your samples. And I know this is a big project, so you're only doing a part, so it may not be.
Let me look.
So the communities that we got the modern, so the subsistence samples from over the, from 2013 to about 2016 were Gamble, Savunga, and Point Lay.
And
Ukqiavik.
And Wainwright.
Okay.
We got samples from Wainwright?
Yeah.
Oh, we got a couple samples from Wainwright. Yeah. So this really was like a pan, effort. We had
the
a lot
project
of-
as a whole had, kind of the archaeological and midden samples and so on. They were from everywhere. So there were Russian sites, there were sites in the Aleutian Islands, in the Bering Sea, all over Alaska. So they really came from everywhere.
Yeah, the archaeological bone was just spread out, from basically Port Moller on the Alaska Peninsula all the way up around the bend to Ukjavik, but also, on the Russian side. So
Port Moller, Moller, as opposed to, since this has been big on teeth, Port Moller.
yes.
Oh, absolutely. So thanks, and I'd like to give a shout out just because it's UAF, and I'd like to give a shout out to the University of Alaska Museum of the North as well. They've got one of the largest in North America, one of the largest mammal collections, and also there's the frozen tissue collection, which has like, I want to say 70,000, over 50,000 specimens, tissues. And so they do a whole, they really keep a great track, and I'm so excited you're working with them. They will continue to archive those, keep those samples, keep the publications all together at the museum. It's really wonderful that you are able to tap into all those efforts of those who came before us. And I'm glad you gave us a little shout out to Bud. And he recognized that really you learn when you're working with the specimens that are coming out of the efforts from the Bering Strait region, as well as the Chukchi. So that was awesome. Thanks for mentioning that. And are you still collecting samples?
No, we're not.
No, we're not.
Okay, because the other thing is, I don't know, we have stranded animals that are a little bit more prevalent than maybe what we've seen in the past. And so I didn't know if there's an effort to talk to Fish and Wildlife Service or people that report strandings, and have, and people are seeing dead walrus. Is that something that is worthwhile for us to be trying to collect when we're
definitely collect it. Absolutely collect it. We're talking about trying to get, another set of funding, follow-up funding, so we can do from 2015, 2016 forward.
Well,
right?
It would be really great to talk to Fish and Wildlife Service to make sure that people who are doing that would be covered. Or maybe Eskimo Walrus Commission, however that would work, they would be covered under, permit paperwork and things like that. So that if you get that, just maybe mention that in, and they can add that to a tally, and you guys could keep sort of passively collecting samples. I don't know how it's working with the harvest community, if you don't have your project. But
so Rafaela is collecting for us still, even though we don't have any any funding to look at things right now, but Rafaela is very much kind of aware, and pretty much any hunter harvested samples that come through her hand, she is sampling.
Yeah, great. Yeah, I was just thinking of the non-harvested carcasses that we see.
If it goes through her hands, then yes.
Yeah, and we're hoping. We also brought back 4 or 5 carcasses in 2016. June 2016,
the epic story.
The epic trip to Point Lay, where we cut up a bunch of walruses that had been trampled on the beach the November before. And the community of Point Lay helped us get out there and helped us get those off the beach.
Into the museum, so they're at
the museum. These full carcasses, 2
and I think
females and 2 babies. One's been put together and has just come back to the museum.
Okay.
So, at any rate, we have those 4 as well. So we have 2 calves and 2 females from that event, and so we've been sort of keeping track. We just. We are going to have to find another pot of money, because none of these crazy chemistry type, isogeochemistry, biogeochemistry type things are cheap.
Yeah.
So
it's a lot of labor of love.
The last thing I'll mention is that the reliance on clams, that's like a Bering Strait thing, Northern Bering Sea. the reliance on maybe a little Bristol Bay, but that's because of the digestion of a lot of the lighter, more flimsy built creatures like polychaete worms and smaller tunicates. Those will go quickly, and so there's a host of animals, and so I just sort of I always hear reliance on clams, and I think one of the things I am always impressed with walrus. I was always taught that in university that the walrus only entered, evolved sort of down in the Pacific in warm water, and then went around the south end of America and came up through the east coast United States, and about 1 million years ago came over the top and entered into Alaska. So if they can do that, I have full faith. They are a smart, strong animal, and I think I have great faith in the walrus and his ability. I'm not saying it's going to be easy what comes next, but
I agree.
We agree. We agree. I think this one takeaway from this project is, it won't be easy, but I felt a lot better at the end of this project than I did at the beginning, right? About walruses and their ability to adapt and survive.
And I would say that goes for the entire Bering Strait ecosystem, including us.
Including the humans, including everyone who's living up there.
That's correct. Yeah.
Mhm.
We're having a lot of things thrown at us, but I think everyone's just going to plan on figuring out the next way forward. So thank you so much. And any other last questions for Laura Horstman or Nicole Misarti? Yes, Rick.
Hi, thanks very much. Really, really interesting. Not so much a question as just a comment on interpretation of the very oldest material. Start to push back older than 3,000 or 4,000 years. The changes in the timing of the seasonality of sea ice might be important with the precession of the equinoxes. So what low ice and high ice means at that kind of time range and before might impact the seasonality of the ice. So just something to keep in mind.
Can you kind of give an
and
example?
also I think sea level was quite different when I think about some of those samples we got from farther south, right? So
a lot of the ice classifications came actually from cores, from sediment
ocean
cores.
cores.
Yeah. So there's a lot of background stuff that we didn't want to bore you guys with. But so there's some background things in there where these classifications came from.
Okay, thanks.
Rick, what do you mean by seasonality? I'm slower than they're smart.
So right now, the sun the Earth is closest to the sun in our winter, Northern Hemisphere winter, and farthest away in our summer. So that has an effect of to some extent balancing out the seasons. That varies on about a 25,000 year cycle.
Oh, okay.
And so about 12,000 years ago, it was exactly reversed. The Earth was closest to the sun in Northern Hemisphere summer and farthest away in Northern Hemisphere winter. And that had the effect of exaggerating the seasons. And so 6,000 years ago, it was spring and fall. And so at those kind of time ranges, it's possible that the seasonality of sea ice, so when the sea ice showed up, when it left for the northern Bering Sea region, may have been different, even if the overall ice appears to be high or low. When it was present and when it wasn't present may have been different because of the difference in the Earth-Sun geometry.
All right, thank you.
I don't know that's the case, but it's just something to keep in mind.
No, it's fascinating. Thank you.
It's the fun of looking so far into the past.
A lot of problems.
All right. Well, any other questions? We should probably let everybody head out. I do want to. I'll say any other questions first. So the next Strait Science, we want to thank Nicole and Misarti and Laura Horseman. And the next Strait Science, we had a cancellation. Coast Guard had to back out, but we are on for the 23rd of September, and there'll be notices going out. And it's going to be, we're going to have Craig Lee. He's with University of Washington, and Sikuliak is headed here. They were up at 79 degrees north just a little while ago, and they're coming back down at the end of this month. And then there's another cruise getting ready to go up. That cruise that's going to switch out here in Nome is going to give a presentation on the 23rd. So we'll learn about what they are planning to do, and hopefully we'll hear from them when they get back as to what they found. That'll be Craig Lee with University of Washington. So